Zona fertilization begins as a single sperm travels through the fallopian tube to reach the ovum. This is the initial step in forming a new life as the sperm prepares to merge its genetic material with the egg.
The sperm and ovum nuclei merge, forming a single-celled zygote. This zygote now contains genetic information from both parents and is ready for its first division.
The zygote undergoes its first cleavage, dividing into two cells. Each division is precise, ensuring proper organization for further embryonic development.
The embryo continues to divide, progressing to 4-cell and then 8-cell stages. These early divisions are crucial for forming a healthy blastocyst.
By day 4, the cells compact tightly to form the morula, a solid ball encased within the zona pellucida, preparing for blastocyst formation.
The morula transforms into a blastocyst, with the inner cell mass destined to form the embryo and the trophoblast to form the placenta, ready for implantation.
The blastocyst travels toward the uterus, maintaining its structure and orientation, ensuring successful attachment to the uterine lining.
The blastocyst attaches to the uterine lining, marking the beginning of implantation. This initial contact is critical for embryo survival.
Trophoblast cells invade the uterine lining, forming the syncytio trophoblast. This ensures the embryo embeds securely and prepares for placenta development.
Spaces called lacunae form within the trophoblast, allowing maternal blood to flow in, supplying oxygen and nutrients to the developing embryo.
Primary chorionic villi develop, extending into the uterine tissue to increase the surface area for nutrient and gas exchange between mother and embryo.
Secondary villi form with an internal mesenchymal core, which will support fetal blood vessel development within the placenta.